Hello everyone. Topic for today presentation is next generation sequencing library, understanding the basic behind each step. So in this lecture, we will be understand the basic principle and a step-by-step workflow of next generation sequencing library preparation. Basically, the goal of library preparation is to convert genomic data into sequencing ready format with adapters and indexes that ensure high quality and reliable sequencing data. So before going into the details, let's try to understand the definition of NGS library. As I mentioned, NGS library is nothing but it is basically preparation of complex DNA into a DNA fragment that could be sequencing ready for the different sequencing platforms. So NGS library is a prepared collection of nucleic acid fragments, whether it could be DNA or cDNA, that had been converted into sequencing ready form by adding platform as specific adapters. So basically it's a smaller DNA fragments obtained from complex genomic DNA, and it should have adapters ligated to both the ends. So adapter have different components like sequencing primer, indexes, and also the flow cell binding sites. So as I understand, genomic library is nothing but is a smaller DNA fragment that has adapter at both ends. So we could easily predict that genomic DNA library preparation or NGS library preparation involves the conversion of complex DNA into a smaller fragment DNA of desired size. So this step is basically achieved by a process called tagementation. So NGS library preparation basically involves these steps like tagementation of genomic DNA, post-tagementation cleanup, and then amplification of tagmented DNA. This is also called index PCR, and then post PCR cleanup is required. So basically that is library purification, and followed by library purification you can say you have prepared the library. So now what is important is once you have library ready is the quantification of the library, and also you have to do the quality check of the library. So now we will try to understand the basic of each steps and what are the precautions that we can take for these performing these. So coming to the first step of the library preparation. So once you have your genomic DNA, for example, if you are preparing the NGS library for your for the DNA sample which have been isolated from the human samples, human WGS. So you have first step is the isolation of genomic DNA and then assessing the quality of the genomic DNA as well as quantity of the genomic DNA. So once you are confident with the quality of the genomic DNA and quantity of the genomic DNA, now you are able to proceed with the genomic library or NGS library preparation. So the first step is tagementation. So tagementation is basically nothing but it is conversion of the genomic DNA into the smaller fragment of the DNA of desired site, and also adding the adapter to the fragmented DNA into a single step. So this is called tagementation. So most of the protocols being used these days is utilizes tagementation for the fragmentation of genomic DNA as well as adding the adapters to the fragmented DNA. rather utilizing the fragmentation steps and adapter ligation steps separately. So the tagementation is achieved by beads link transposes that cleaves the DNA and also add the adapters to the fragment DNA into one step. So at the end of the tagementation process, what you have is a smaller DNA fragment of desired size having the adapter sequences at both the ends. So this is basically outcome of the tagementation process. So tagementation is basically done at the advised temperatures in the commercially available reagent kits. So basically you have to use the temperature. Mostly it is done at 55 degree centigrade for 15 minutes or maybe at 37 degrees. So depending on the protocol which you are using. So post tagementation, what is important is post-fragmentation cleanup. So basically this steps stabilizes the library by stopping the enzymatic activity, that is stopping the transposase activity and removing unwanted components before you go for the next step, that is amplification of the segmented DNA. So basically the key purposes of this post-fragmentation cleanup is terminate the tagementation reaction so that there should be no over tagementation that will stop the tagementation process, prevent further fragmentation, and also to remove the transposes enzyme so that there will be no interference into the downstream process. So if I'll tell you the methodology, so generally once the tagementation is over, you just take the tagementation reaction from the thermal cycler, and what is important is you have to add tagementation stop buffer which is being provided into the commercial available kit, and then again you have to incubate the reaction for the advised duration at advised temperature, and then you have to proceed with the washing. So that washing basically removes the in residual enzymes like transposes and also the salts and all, and the uh, making the fragments clean. And what is important is it also eliminates the post segmentation cleanup, also eliminate the free untagged adapters. So at the end, you will have clean, stable amplification ready DNA library. So coming to the next step of the library preparation is amplification of the fragmented DNA. That is also called index PCR. So basically, these steps is increasing the quantity of the library that would be sufficient for the sequencing platform, and also it adds the unique indexes. So these indexes are commercially available. So these steps basically amplify the adapter tag DNA fragments, so that will ultimately increase the quantity of the library that could be sufficient for the sequencing platform, and also it added adapters so that you can pool multiple samples together. Basically this will facilitates the multiplexing of the samples, and later once the sequencing is done, sequencing runs complete. During the analysis, using the sequencing of the sequences of the indexes, you can do the demultiplexing, that is you can separate the data for each sample, and also it will give you basically at the end of this process you will have the sequencing library structure. So now once you have the this step is, again the purification is required. So basically this is called cleanup of library, or you can say post PCR cleanup. So basically this step removes the unwanted PCR components like DNTP, salts, enzyme, and so this remove the PCR byproducts, and also the size selection, which is very important. The all if your desired size of the library is, for example, 400 to 500 base pair, it will select based on the magnetic beads which you are going to use for the purification, and the larger size or smaller size fragments of the DNA will be excluded, and also the dimers and all will be excluded. So this step will ensure the uniform library fragment distribution, and also it will improve the overall library purity and will give you the sequencing ready DNA fragments. So once you have your library, it is very important to quantify the library, because this will this based on the quantification you will be able to decide the loading concentration for the sequencing to the sequencing platform. There should be no overload, there should be no underload, because it will impact very badly the sequencing and the data generation. So basically this step is important because it measures the accurate library concentration and determine the amount of usable DNA, and it will prevent over or underloading during sequencing, and also it will help you during pooling, so that you will have equal representation of all the sample based on the concentration which you are going to multiplex during the sequencing, and based on the concentration, equal concentration only, you will be able to achieve the optimal cluster density that will maximize the data quality and yield. So at the end, so library, basically library quantification ensure that correct amount of DNA is loaded for sequencing, which is very critical for achieving optimal data distribution across the sample. So now very important is library quality check. So now everything is okay. You have your library, the concentration is good. So you can do the library quality check. So, so that you will be able to proceed with the high quality library. So this is basically based on the size of the library. For example, if you have made your library aiming the size 400 to 500 base pairs, so based then you can analyze your library using different equipments. If you have devastation in your laboratory, you can use devastation, or you can use fragment analyzer from Agilence, or also if you have QSEP 100, you can use QSEP. So any of these uh, equipment can be used for the quality check of the library. So basically this step will assess the library for fragment size distribution. So what is important is here you should see a single peak of desired size. So as graph it's shown here, you can see a single peak. If you are observing multiple peaks of same intensity, or the height of your peak is very low, in that case you have to doubt your library, or you should exclude that sample for putting for the sequencing run. So basically this step ensure the integrity and purity of the library, and it also confirm the sequencing readiness. So putting all the steps together, basically the NGS library preparation is nothing but it is a conversion of complex genomic library into a fragmented DNA that will have adapters, that should have indexes so that you can multiply, and at the end it will be able to point with the sequencing flow cell. So thank you for now. In my next video, I will be explaining the techniques, the technicality and methodology behind each step. Thank you. Thank you very much.